Number 1. The Jack Hill Zircons. Australia, approximately 4.4 billion years old. Picture Earth roughly 100 million years after it formed. The planet is young, unstable, and still cooling from its violent assembly. The surface is being battered by debris left over from the formation of the inner solar system. For most of the 20th century, this period, known as the Hadean Eon, was imagined as a hellscape of lava oceans and constant impact. The name Hadean itself comes from Hades, the underworld of Greek mythology. That image, however, has been quietly overturned by some of the smallest objects geologists have ever studied. Tiny crystals of zircon, recovered from the Jack Hills region of Western Australia, return ages of about 4.404 billion years, with an uncertainty of plus or minus 8 million years. These grains are older than nearly anything else on Earth. They are not whole rocks. They are individual mineral crystals smaller than a grain of beach sand, locked inside much younger sedimentary rocks called conglomerates. The host rocks are roughly 3 billion years old, but the zircons inside them are far older. They were eroded out of vanished early crust, transported, redeposited, and preserved by accident. To understand why these grains matter, you need to know what zircon does at the atomic level. When zircon crystallizes, it readily accepts uranium atoms into its structure but rejects lead. Over hundreds of millions of years, that uranium decays into lead at a known rate. The crystal becomes a self-contained radiometric clock. Geologists measure the ratio of remaining uranium to accumulated lead and read the age. Zircon is also extraordinarily durable. It survives erosion, burial, intense pressure, metamorphism, and crustal recycling that destroys almost everything else. The most provocative clue, though, is not the age itself. It is the oxygen isotope ratio inside the crystals. Certain elevated ratios, particularly enriched oxygen-18 relative to oxygen-16, suggest that the parent magma had interacted with material altered by liquid water at low temperatures. The wording matters. The zircons suggest evidence for liquid water. They do not guarantee global oceans or stable shorelines. Still, the chemistry implies that Earth had cooled enough for crust and water-rock interaction far earlier than the old hellscape model permitted. Some grains also show a chemistry consistent with differentiated granitic-style continental crust, the kind of lighter rock that floats above the denser basaltic plains forming most ocean floor today. If that interpretation holds, the very ingredients of continents may have been present within the planet's first 150 million years. The moon-forming impact chronology is genuinely complex, so it would be wrong to simply call these crystals older than the moon. What can be said is that they are fragments of a planet whose entire surface no longer exists. The original Hadean crust was destroyed by plate recycling and bombardment. These zircons are eyewitnesses without a face, miniature time capsules carrying chemistry from landscapes that vanished before anything resembling a continent in the modern sense had stabilized. Modern researchers use these grains to reconstruct vanished planetary conditions, atmospheric composition, and possible early habitability windows. A grain smaller than a poppy seed, then, may carry evidence of Earth's first wet crust. So if minerals can preserve time because physics locks radioactive decay into a crystal structure, what happens when biology starts trying to do the same thing with bodies instead of crystals? Number 2, the Francevillian Biota. Gabon, approximately 2.1 billion years old. Move forward roughly 2 billion years, the planet is no longer a recently assembled object, continents have come and gone. Microbial life has been processing sunlight and chemistry for over 1 billion years. This is the Paleoproterozoic, long before animals, plants, or any visible ecosystems exist. Yet in black shales from the Francevillian Basin in Gabon, paleontologists have found something that, if biological, scrambles the expected order of life's history. Dating to about 2.1 billion years, these structures are centimeter scale, which is enormous for that age. They appear after the Great Oxidation Event, the period when atmospheric and oceanic oxygen rose dramatically due to photosynthetic microbes. Oxygen matters for one straightforward reason: larger bodies require more energy. Aerobic metabolism, the kind that uses oxygen, releases far more energy per unit of food than anaerobic metabolism. With more oxygen available, the energetic ceiling for body size rises. The Franceville structures take advantage of that ceiling, at least if you accept the biological reading. They are flattened discs, lobate shapes, and forms with radial organization. Some specimens reportedly reach around 12 centimeters across. To compare, that is roughly the size of a modern tea saucer, formed nearly 1.5 billion years before the first widely accepted animals. The original interpretation, published in 2010 by a team led by Abderrazak El Albani at the University of Poitiers, proposed that these were colonial organisms living in oxygenated marine environments. Preservation came partly through periodization, the replacement of soft tissue or its outline by iron sulfide, which forms reflective golden mineral textures. Black shale itself is a useful preservation environment. Fine sediment, low oxygen at the seafloor, and slow burial can capture delicate shapes that would normally rot away. Subsequent work has tried to push the question further. Trace metals and zinc isotope studies have been used to test whether the structures show chemical signatures consistent with biological metabolism. The mainstream interpretation today is cautious. These may be macroscopic colonial organisms, but they are not confirmed animals, and they are not necessarily even eukaryotes in the modern sense. Some researchers question whether the structures are truly biological at all, suggesting they could be products of inorganic processes acting on layered sediments. That debate is unresolved. What makes the Francevillian Biota a genuine anomaly is timing. They appear roughly 1.5 billion years before the Ediacaran fauna that most textbooks treat as the first major experiment in large body size. If they are biological, they may represent a local oxygenated ecosystem that flourished briefly and then disappeared, leaving no obvious descendants. That possibility carries a heavy implication. Complex life may have evolved, collapsed, and had to re-evolve much later under different conditions. The classical narrative of complexity climbing a staircase from microbe to animal becomes a landscape of attempts. Most failed. One eventually did not. Modern relevance is direct. Astrobiologists searching for early life on Mars or in distant exoplanet atmospheres are now thinking carefully about what biosignatures from a brief extinguished biosphere would even look like. If we cannot confidently read Earth's own record, reading another planet's becomes harder still. So if complex life experimented and vanished once, what about the next great burst, the one that left fossil seafloors on multiple continents and bodies that look like nothing alive today? Number 3. The Ediacaran Biota, Dickinsonia and Charnia, approximately 570 to 558 million years old. The Ediacaran period runs from roughly 635 million years ago to about 541 million years ago. It sits just before the Cambrian explosion, the burst of recognizable animal body plans that produced trilobites, early arthropods, and the deep ancestors of nearly every modern animal group. The Ediacaran is older, stranger, and in many ways harder to read. Most familiar animal groups had not yet appeared in the fossil record. What had appeared were soft-bodied organisms preserved as impressions in sandstone, often across entire bedding plains that look like fossil seafloors. Two of the most discussed forms are Dickinsonia and Charnia. Dickinsonia is oval, ribbed, flattened, and soft-bodied. Specimens range from a few centimeters to over 1 meter long. It has no obvious head, no limbs, no eyes, no skeleton, and no preserved digestive organs. For decades it puzzled paleontologists. Was it an early animal, a giant single-celled organism, a fungus, or something entirely outside modern classification? Charnia, first described from Charnwood Forest in central England, is quilted and frond-like. It rises from the seafloor on a holdfast, the anchoring base, and extends a leaf-shaped body upward. It is not a plant. Some Charnia specimens lived in deep marine settings far below the photic zone, the depth where sunlight can still drive photosynthesis. Whatever Charnia was doing, it was not relying on light. It also displays glide reflection symmetry, where body segments offset along a midline rather than mirror each other. Modern animals overwhelmingly use bilateral or radial symmetry. Glide reflection is rare. The interpretive breakthrough for Dickinsonia came from molecular paleontology. In 2018, researchers led by Ilya Bobrovsky, then at the Australian National University, extracted ancient organic molecules from exceptionally well-preserved Dickinsonia specimens in Russia. They found cholesterol-like steroid biomarkers. Cholesterol is a sterol overwhelmingly associated with animal tissues. That chemical signal is among the strongest evidence yet that Dickinsonia was an animal in some real sense, not a fungus, not an alga, not a colonial protist. Most Ediacaran organisms lived on or within microbial mats, the slimy living surfaces that coated late Precambrian seafloors. Preservation took the form of impressions in sandstone rather than mineralized hard parts. Soft-bodied fossilization required unusual sediment in microbial and chemical conditions. The whole Ediacaran fossil window exists because ocean chemistry and microbial communities behaved in ways that no longer happen at large scale. By the start of the Cambrian, that preservation mode largely disappears. Burrowing animals churned the sediment. Predators with claws and shells appeared. Microbial mats were broken up. The Ediacaran world stopped being recordable in the same way. Many Ediacaran forms vanish near the Cambrian boundary. Some lineages, including Dickinsonia, seem to leave no obvious descendants. Researchers have called this a possible failed kingdom, a branch of large-bodied life that briefly succeeded and then was outcompeted, eaten, or destabilized. The Cambrian explosion was not the beginning of animals. It was a reorganization of ecosystems, with skeletons, eyes, burrowing, and active predation reshaping which body plans could survive. Not every Ediacaran lineage necessarily failed, and some may have given rise to surviving animal groups. The picture is mixed. What stays clear is that biology explored forms before settling into the patterns we recognize. So if soft bodies leave such fragile traces, what does it look like when an entire ecosystem is preserved not as scattered impressions, but as a buried landscape sitting above your head? Number 4. Springfield Coal Mine Fossil Forest. Illinois, approximately 307 million years old. The Pennsylvanian Carboniferous, around 300 to 7 million years ago, predates the first dinosaurs by roughly 75 million years. To make that comparable to a human scale, it is more than 300 times the age of the entire genus Homo. In the Springfield coal member of the Illinois Basin, underground coal mining has exposed something rare in the fossil record. Not a leaf, not a stump, an entire fossil forest, preserved in 3 dimensions across square kilometers of mine roof. Coal miners working the Springfield seam look upward and see the underside of an ancient forest floor. Tree trunks intersect overhead. Root systems spread sideways through the rock. The shale that forms the mine roof is, in effect, a buried wetland surface frozen in place. The coal seam below it represents compressed ancient peat, the dense organic material that accumulated when plant matter could not fully decay. The forest appears to have died and been buried suddenly. Researchers, including teams led by Howard Falcon Lang and William DeMichel, have linked the rapid burial to earthquake-driven subsidence where a section of land dropped into water and was sealed under sediment before normal decay could destroy it. Rapid burial matters because forests almost never preserve standing. Roots rot, trunks fall, leaves decompose, and the ecosystem turns to dust. Quick sealing under fine sediment locks the forest in spatial position. Major plants in the assemblage include lycopsids, tree ferns, seed ferns, and calamites. Lycopsids in this period included giant forms such as Lepidodendron, towering relatives of the small club mosses still alive today. They reproduce by spores rather than seeds and could exceed 30 meters in height. Calamites were tree-sized horsetails segmented and hollow-stemmed. Tree ferns and seed ferns formed lower canopy and understory layers. Many of these plants reproduced by spores, a method that ties them tightly to wet environments. The Carboniferous atmosphere itself was different. Atmospheric oxygen levels are widely modeled to have been substantially higher than today, possibly above 30% compared with the current 21%. High oxygen supported giant insects including dragonfly relatives such as Meganeura, with wingspans reaching 70 centimeters. It also supported intense coal formation because vast amounts of plant material accumulated in low-decay swamps. Fungi capable of breaking down lignin, the tough polymer in wood, may not yet have been efficient enough to keep up. The mainstream interpretation of the Springfield site is straightforward. It is a buried peat-forming tropical wetland near the paleo-equator, preserved by sudden subsidence. The archaeological angle, in a geological sense, is the spatial preservation. The mine roof preserves ecological zoning across an ancient swamp, with different plant communities in different microenvironments. You can map a vanished landscape simply by looking up. The more speculative reading is the time capsule effect. A whole vanished climate is still spatially intact, not just inferred. Modern relevance is layered. Fossil forests help reconstruct paleoclimate, atmospheric oxygen, and the formation of coal that fueled industrialization. The same coal that powered the early factories of the 1800s began as a swamp that died in a single, rapid event hundreds of millions of years before any vertebrate walked on land. If sediment, water, pressure, and time can preserve an entire ecosystem, what kinds of behavior can stone preserve, and how far back does that record really go? Number 5. Lomekwi stone tools. Kenya, approximately 3.3 million years old. Most people, asked when toolmaking began, picture a cave-dwelling early human-shaping flint. The reality is older, stranger, and not necessarily human at all. At a site called Lomekwi 3, near the western shore of Lake Turkana in Kenya, archaeologists have recovered stone tools dated to about 3.3 million years ago. The site was reported in 2015 by a team led by Sonia Harman. The dating predates the Oldowan tradition, the earliest previously recognized stone tool industry, by about 700,000 years. It also predates the earliest known fossils of the genus Homo by roughly 500,000 years. That gap is critical. Whoever made these tools was not Homo as we usually define the term. Possible candidates include Kenyanthropus platyops, known from the same general region, or Australopithecine relatives. These were small-brained, upright walking hominins with ape-like features and some human-like adaptations. The implication is that pre-Homo hominins were already breaking stone with intention. The Lomekwi assemblage includes cores, the original blocks of stone from which flakes are struck, along with flakes themselves, anvils, percussors, and worked cobbles. The reported counts include approximately 83 cores and 35 flakes among the recovered pieces, with the assemblage continuing to grow as fieldwork proceeds. The artifacts are large and heavy compared with later Oldowan tools. Some weigh several kilograms. Stone knapping, the controlled fracture of rock to produce sharp edges, is not random smashing. It requires understanding of how brittle stone behaves under impact. Strike a core at the wrong angle and you get useless rubble. Strike it correctly and a usable flake detaches with a sharp edge. The Lomekwi pieces show battering marks, flake scars, and pounding damage consistent with deliberate percussion, not natural breakage. Raw materials include basalt, phonolite, and trachyphenolite, volcanic rocks available in the region. This is not the fine-grained flint of European caves. The makers were using what was locally abundant. The paleo environment around Lomekwi at that time appears to have been wooded, with trees and shrubs rather than open grassland. That detail matters because earlier theories tied tool use to savanna expansion. The idea that drying climates pushed hominins onto open plains where they needed tools to butcher game. Lomekwi suggests stone-breaking behavior emerged in a more closed, wooded environment, possibly for cracking nuts, processing plants, or other tasks unrelated to hunting large animals. The mainstream interpretation is that early hominins had stone-breaking skill before Homo existed. The Stone Age, in other words, may begin outside the Homo-centered timeline. The more speculative reading is that the cognitive roots of technology are much older and more widely distributed among hominins than previously assumed. There is no clear evidence that the Lomekwi tools were used for systematic butchery of large animals. The maker's behavior, language, and culture did not survive. The stone survived. So if technology can persist while its makers vanish, what about a single individual organism that has not vanished, but has lived continuously through human prehistory and into the present? Number 6. Pando, the Trembling giant. Utah, possibly 16,000 to 80,000 years old. In Fish Lake National Forest in central Utah grows a stand of quaking aspen that, at first glance, looks like a normal forest. White bark trunks rise vertically, leaves flutter in the wind. Hikers walk through it without realizing what it is. The stand is called Pando, Latin for eye spread. Pando is not a forest. It is a single male quaking aspen, a clonal organism whose trunks all share one underground root system. The above-ground area of Pando covers about 106 acres. Its estimated mass is nearly 13 million pounds, or roughly 6 million kilograms. It is widely described as one of the heaviest known organisms on Earth. Stem counts vary, but estimates often cite around 47,000 individual trunks rising from the same root network. Each trunk is not ancient on its own. Aspen trunks typically live for 100 to 150 years. The ancient body is the root system underneath. Aspen reproduces by suckering, sending up new vertical stems from horizontal roots. When an old trunk dies, new ones replace it from the same genetic individual. Pando looks like a forest, but functions genetically as one organism. Every leaf, every trunk, every branch shares the same DNA. Estimating the age of a clonal organism is genuinely difficult. The popular figure of 80,000 years is best treated as a high-end estimate, derived from modeling of growth rates and clonal expansion. The broader scientifically modeled range is roughly 16,000 to 80,000 years. Even the lower end places Pando well before the rise of agriculture, which began roughly 12,000 years ago. The upper end places it before the last glacial maximum, before the first known cave paintings of Lascaux, before nearly every cultural marker of human history. Pando survived major post-Ice Age climate swings, shifts in regional water availability, and the entire span of human civilization within its range. It did so without memory, language, or movement. It did so by repetition, regenerating its body part by part across millennia. The mainstream interpretation is that Pando is an ancient clone maintained by repeated regeneration, not by any single trunk's longevity. The historical ecology angle highlights its resilience. The more speculative reading is that life can preserve itself through repetition when the surrounding ecosystem stays within tolerable bounds. Most of Pando's history is biological continuity without any written record. Pando today faces real threats. Mule deer browsing, cattle grazing, drought stress, and poor recruitment of new stems are reducing its capacity to regenerate. Researchers including Paul Rogers at Utah State University have documented declining young stem survival in unfenced areas. Fenced sections of the clone show better recovery because young stems are protected from browsing pressure long enough to mature. The implication is that Pando's continuity depends on regeneration, not on the survival of any single visible trunk. Conservation must protect the process, not just the photogenic stand. Life can outlast civilizations when roots, sunlight, water, and the rules of cloning align. So if a living organism can persist through all of human prehistory, what kind of cultural mark survives the same span, and who left it? Number 7. Maltravieso hand stencils. Spain, approximately 64,000 to 66,000 years old. Maltravieso cave sits in the city of Cáceres, in the Extremadura region of western Spain. Inside its passages, on cave walls, are red ochre hand stencils. The technique is intimate and easy to picture. A person places a hand against the rock. Pigment, usually mixed with water or saliva, is then blown or sprayed around the hand, often through a hollow bone or reed. When the hand is removed, a negative silhouette remains, the hand outlined by pigment but unpainted itself. Some Maltravieso stencils are dated to more than 66,000 years old. The broader Iberian cave art dating effort, published in 2018 by a team led by Dirk Hoffman, returned ages older than 64.8 thousand years for selected pigment samples in 3 Spanish caves, including Maltravieso, La Pasiega, and Ardales. The dating method is uranium-thorium analysis of thin calcite crusts that formed over the pigment. Calcite, calcium carbonate deposited by mineral-rich water dripping inside caves, accumulates slowly. By measuring the ratio of uranium to its decay product thorium within the crust, researchers can establish when the crust formed. Because the crust sits on top of the pigment, the crust must be younger. The pigment must therefore be older than the calcite covering it. This method gives a minimum age. The painting itself could be older than the crust by an unknown amount. The implication is sharp. 66,000 years ago, anatomically, modern humans are not securely documented in Western Europe. The earliest secure modern human evidence in the region falls roughly 20,000 years later. The likely authors of these stencils, by elimination and by direct association at related sites, are Neanderthals. Red ochre is iron-rich mineral pigment, primarily hematite, ground and mixed with binders. Producing a hand stencil requires planning, pigment preparation, deliberate bodily positioning, and the symbolic intent to leave an enduring mark. These are not survival behaviors. They are cultural ones. The mainstream interpretation, as advanced by the Hoffman team and supported by other evidence of Neanderthal symbolic activity, is that Neanderthals made symbolic marks and possibly cave art. Neanderthals, in this view, were not merely survival machines. They had culture, planning, and aesthetic behavior. The more speculative reading is that symbolic thought may be a shared inheritance of multiple human species, not something that emerged uniquely with Homo sapiens. The debate is real. Some specialists question the calcite dating, raising concerns about uranium movement within porous rock, possible contamination, or sample integrity. Others accept the dates but argue the artwork is too ambiguous to assign confidently to Neanderthals. The careful wording is attributed to Neanderthals, not proven beyond dispute. Neanderthal culture vanished. They left no writing, limited preservation, and a fragmented archaeological record. What survives at Maltravieso is pigment on stone, locked under a calcite layer that ticks like a uranium clock. Symbols emerge when mined, mineral pigment, and a surface become part of the same act. So if a stencil is a symbol, what does it look like when symbols start telling stories with figures interacting in scenes? Number 8. Sulawesi hunting scene. Indonesia, at least approximately 51,200 years old. For decades, the standard story of art's origin centered on Europe. Lascaux in France, Altamira in Spain, the cave bears and bison and aurochs of the late Pleistocene Cordillera were treated as the high point of early human creativity. Discoveries on the island of Sulawesi in central Indonesia have rewritten that geography. Limestone caves in the Maros Pangkep region preserve cave art dating to deep Ice Age prehistory. In 2019, a team including Maxime Yobert, Adam Broom, and colleagues reported a hunting scene with an initial minimum age of about 43.9 thousand years. Subsequent work using improved laser ablation uranium series dating, published in 2024, returned older minimum ages for related Sulawesi panels, including dates of roughly 50.2 plus or minus 2.2 thousand years and 53.5 plus or minus 2.3 thousand years. One particular composition has been pushed to a minimum of about 51,200 years. The image shows human-like figures interacting with wild pigs, the latter most likely Sulawesi warty pigs. Some figures are interpreted as therianthropes, hybrid forms combining human and animal features. Therianthropic imagery is significant because it implies imagined beings, not direct observation. It is a window into mythic or symbolic thought. The pigment is red mineral colorant applied to limestone walls. The dating method targets calcium carbonate crusts that form on the painted surface. As at Maltravieso, this method establishes minimum ages. The actual paintings could be older. Laser ablation uranium series dating refines this approach by sampling tiny spots within the crust, allowing dates to be tied tightly to specific pigment locations rather than averaged across larger samples. What sets the Sulawesi hunting scene apart is narrative. Multiple figures appear to interact in a shared scene. This is different from isolated handprints, abstract marks, or single animal figures. Animals in the scene are rendered more naturalistic than the human-like figures, a pattern also observed in some European Paleolithic art. The mainstream interpretation is that this is among the oldest known narrative art. The historical implication is that early symbolic culture was not limited to Europe, was not invented by a single population, and may instead reflect cognitive capacities present across widely dispersed Homo sapiens groups. The more speculative reading is that mythic imagination, the ability to invent beings that do not exist and depict them as if they do, may have emerged very early in human dispersal across Asia. Pigment survived only where cave chemistry protected it. Limestone caves, mineral crusts, controlled humidity, and luck combined to preserve what would otherwise have flaked off thousands of years ago. The surface decay observed in Sulawesi today, including exfoliation of pigmented layers driven by climate change and seasonal humidity shifts, suggests that some of the earliest evidence may be deteriorating faster than it can be documented. The exact meaning of the hunting scene remains unknown. Whether it depicts a real hunt, a myth, a ritual, or something else cannot be recovered. What is clear is that storytelling appears to begin when perception, memory, pigment, and shared attention become visible at the same time. So if storytelling can survive 50,000 years in pigment, what can survive across thousands of years in flesh and bone on the other side of the world. Number 9. Wrangel Island Pygmy Mammoths. Arctic Russia, survived until approximately 4,000 years ago. Wrangel Island sits in the Arctic Ocean, north of eastern Siberia, separated from the mainland by the Chukchi Sea. It is roughly the size of the Hawaiian Big Island, mountainous, treeless, and one of the harshest environments humans have ever set foot on. It is also the place where a woolly mammoths made their last stand. Mainland populations of woolly mammoths, Mammothus primigenius, largely vanished near the end of the Pleistocene, around 10,000 to 11,000 years ago, as climates warmed and tundra ecosystems contracted. Most of the species was gone. On Wrangel Island, however, an isolated population persisted until roughly 4,000 years ago. The implication, when placed against human history, is striking. 4,000 years ago is well after the development of agriculture in multiple regions, well after the first cities of Mesopotamia, well into the Bronze Age. The Great Pyramid of Giza, attributed to the pharaoh Khufu, dates to the early 25th century, before the Common Era, approximately 4,500 years ago. That places Khufu's world and the last living mammoths on the same broad calendar. Pyramids and mammoths overlapped. The Wrangel mammoths were smaller than many mainland mammoths, leading to the popular shorthand Pygmy mammoths, though that term is not always treated as taxonomically precise. Island isolation often produces dwarfism in large mammals through a process known as island dwarfism, where limited food and reduced predator pressure favor smaller body size. Genetic studies, including work led by Love Dahlin and colleagues at the Center for Paleogenetics in Stockholm, show that the Wrangel population went through severe genetic bottlenecks. With only a few hundred individuals at most, inbreeding became unavoidable. Earlier research suggested signs of genomic meltdown, the accumulation of harmful mutations that small populations cannot purge. More recent genomic work, however, has argued that the population remained relatively stable for many generations after the bottleneck, with no clear pattern of accelerating decline up to the moment of extinction. The mainstream interpretation is that extinction likely involved environmental stress, vulnerability of small populations to chance events, or a sudden ecological disruption. Possible final triggers include disease, extreme weather such as a rain-on-snow icing event that locked vegetation under impassable ice, food collapse, or some combination. Humans appear to have arrived on Wrangel Island only after the mammoths were already gone, based on current archaeological evidence. So unlike many island megafauna extinctions, this one is not obviously linked to direct hunting. The more speculative reading is that the last mammoths may have died from a sudden, unlucky ecological accident rather than a slow decline. The final death left no written witness, only bones and DNA preserved in permafrost. Modern relevance is direct. Island populations today, from the Channel Island fox to the Florida panther to many endemic species worldwide, face similar bottleneck and climate risks. Conservation genetics is essentially the science of preventing what happened on Wrangel from happening again. Extinction often happens when climate, isolation, genes, and chance converge. So if mammoths could persist in flesh until the age of pyramids, what living animal alive right now could carry comparable historical depth inside its body? Number 10. The Greenland Shark. Lifespan approximately 272 to over 500 years. In the cold, dark waters of the North Atlantic and the Arctic, beneath ice shelves and along deep continental slopes, swims a slow, enormous, almost mythological animal. Somniosus microcephalus, the Greenland shark, is the longest-lived known vertebrate. The species name microcephalus refers to its proportionally small head. Somniosus comes from a Latin root meaning sleepy or drowsy, fitting an animal that moves at less than 1 mile per hour even when actively swimming. Greenland sharks are deep, cold-water sleeper sharks. They can be found in waters from the surface in cold regions down to nearly 2,000 meters. Adults can exceed 4 to 5 meters in length, with some specimens documented over 6 meters. They grow extraordinarily slowly, often around or below 1 centimeter per year. The longevity of Greenland sharks could not be measured by traditional methods. Most shark species are aged using vertebral bands or other calcified structures that lay down growth rings, similar to tree rings. Greenland sharks lack useful hard aging structures. Their cartilaginous skeletons do not preserve a clean record. The breakthrough came from an unexpected place: the eye. The protein crystallines in the lens nucleus of a vertebrate eye form during early development and remain metabolically stable throughout life. They are, in effect, a tissue clock laid down at birth and never reset. In 2016, a team led by Julius Nielsen, then a doctoral student at the University of Copenhagen, applied radiocarbon dating to eye lens proteins from 28 female Greenland sharks. The team used the Cold War bomb pulse calibration. Atmospheric nuclear weapons testing in the 1950s and early 1960s released a sharp spike of carbon-14 into the atmosphere. That spike was absorbed into ocean food webs and into the proteins of any organism alive at the time. Sharks born after the spike show the elevated signal in their lens nuclei. Sharks born before do not. By measuring radiocarbon signatures and applying growth modeling, the team established a minimum lifespan of at least 272 years. The two largest females studied returned estimated ages of approximately 335 and 392 years. The full uncertainty range for the oldest estimate spans roughly 272 to 512 years. The mainstream interpretation is that slow metabolism, cold water temperatures, and slow growth all contribute to extreme longevity. Cold slows biological processes. Slow growth reduces accumulated cellular damage. Low predation pressure in deep water removes a major source of mortality. The historical angle is genuinely strange. One of the largest females in the Nielsen study could have been born before Isaac Newton, who entered the world in 1642. A single living shark may have been swimming when the first telescopes were aimed at the moons of Jupiter. The more speculative reading is that extreme longevity may hide biological repair mechanisms not yet fully understood. Comparative biologists are now studying Greenland sharks alongside bowhead whales and rough-eye rockfish to understand how some vertebrates resist the normal accumulation of cellular damage with age. The modern conservation risk is direct. Greenland sharks reach sexual maturity at approximately 150 years of age. A species that takes a century and a half to reproduce cannot recover quickly from population losses. Bycatch in commercial fisheries, long misunderstood as low impact, may be removing centuries of growth in a single haul. Conservation must think in centuries, not seasons. Longevity is biology obeying the slow mathematics of metabolism, growth, and cold. So if minerals, fossils, forests, tools, pigments, ecosystems, and living organisms can all carry deep time inside them, what does it mean that none of these timelines line up the way the textbook story would expect? Each anomaly in this documentary punctures one assumption about how the past should behave. Together, they suggest a deeper pattern. Deep time is not experienced as a clean ladder. Earth history is reconstructed from surviving fragments, never from a complete archive. The fragments survive only under extreme conditions. Zircons survive because their crystal structure resists almost everything. Cave art survives because calcite crusts and dry chambers preserve pigment for tens of thousands of years. Pando survives because aspen suckering bypasses the death of individual trunks. Greenland sharks survive because cold water and slow metabolism stretch a single life across centuries. Each surviving record is a different kind of accident. Fossil and archaeological records are biased by preservation. What survives is not always what was most common, most important, or most representative of its time. Hard-bodied organisms survive better than soft ones. Pigment under calcite survives better than pigment exposed to weather. Stone tools survive better than wooden ones. Mineralized teeth survive better than skin. Many firsts claimed in headlines are only the earliest evidence found so far. They are placeholders waiting for older finds. The Lomekwi tools displaced the Oldowan as oldest known. Sulawesi displaced parts of Europe in the cave art timeline. Future discoveries are likely to displace these in turn. The mainstream principle is that timelines shift because better methods find older evidence. The historical pattern is that many discoveries arise from accidents. Mining exposed the Springfield fossil forest. Cave exploration revealed Maltravieso and Sulawesi. Erosion brought zircons to the surface in Western Australia. The more speculative possibility is that much of Earth's early complexity existed and left no trace at all. Oldest known does not mean first ever. Minimum ages, common in cave art and certain fossil contexts, mean only that an event happened before a measurable cap. The actual event could be much older. The dating tools themselves shape scientific narratives. Uranium-lead dating opened deep time for minerals. Uranium-thorium dating made minimum ages possible for cave art. Radiocarbon dating opened the late Pleistocene and Holocene. Genetic modeling extended timelines for organisms that lack hard parts. Bomb pulse radiocarbon allowed Greenland shark longevity to be measured at all. Modern relevance is straightforward. The public hears headlines while scientists work in probability ranges. Time becomes knowable only when matter preserves a measurable signal. A central question runs through every anomaly in this documentary. Does complexity build steadily, disappear, and re-emerge, or erupt in separate, disconnected bursts? 3 broad theories address this question, each with intellectual merit. The first is mainstream gradualism. In this view, complexity accumulates through environmental change, genetic innovation, and selection over long timescales. Oxygenation drives larger bodies and active metabolisms. The Francevillian structures, if biological, fit this model cautiously as possible early complexity following the Great Oxidation event. Ediacaran life fits as later, large body experimentation that set the stage for Cambrian ecosystems. Lomekwi stone tools fit as behavioral complexity emerging gradually within hominin lineages, not as a sudden human invention. Each step builds on what came before, even when individual lineages fail. The second theory is the lost experiment model. In this view, evolution repeatedly tries complex forms, some of which vanish without leaving direct descendants. Francevillian Biota becomes a possible early experiment in macroscopic complexity that did not lead directly to animals. Ediacaran body plans become large-bodied attempts that disappeared near the Cambrian boundary, replaced by faster, harder, more predatory ecosystems. Wrangel mammoths become a remnant from a vanished Ice Age system, surviving briefly into a warmer world before dying out. The lost experiment model takes seriously the possibility that the history of complexity is a record of attempts, most of which fail. The third theory is the speculative hidden complexity model. In this view, much more early complexity may have existed, but the evidence has been erased. This is not an argument for hidden civilizations or fantasy biology. It is a reasoned framework grounded in physical evidence. Soft-bodied organisms decay easily. Symbolic cultures leave no surviving language. Erosion, tectonics, and sea level change destroy ancient surfaces. Wooden tools rot. Pigments wash off. The hidden complexity model holds that fragile evidence is easily destroyed by ordinary processes, and that the gaps in the record are themselves data. Speculation must stay tied to physical evidence. Without that constraint, the framework becomes guesswork. Each evidence type answers a different question. Mineral chemistry answers questions about early Earth conditions. Body fossils answer questions about anatomy and ecology. Tools answer questions about behavior. Art answers questions about cognition and culture. DNA answers questions about lineage and population history. Living organisms like Pando and the Greenland Shark answer questions about durability. New methods can revive old debates without inventing unsupported claims. Improved isotope analysis, ancient DNA recovery, laser ablation dating, and genomic modeling are all reshaping debates that had seemed settled. Complexity is not one staircase. It is a landscape of experiments shaped by available energy, environment, and accident. Every anomaly in this documentary depends on a clock hidden in matter. Different clocks measure different timescales, and each method has strengths and limitations. Uranium lead dating is central to the Jack Hill Zircons. Uranium decays into lead at a known rate over billions of years. Zircon is ideal for this method because uranium fits readily into the crystal structure during formation, while lead is largely excluded. Any lead found later is therefore the product of decay, not initial contamination. The method works best for samples older than about 100 million years. Uranium-thorium dating is useful for cave calcite crusts at sites such as Maltravieso, La Pasiega, Ardales, and Sulawesi. Calcite forms slowly from mineral-rich water. By measuring the ratio of uranium to its decay product thorium, researchers establish when the crust formed. Calcite over pigment provides a minimum age because the pigment must be older than the layer covering it. The actual painting may be considerably older. Radiocarbon dating works for carbon-bearing material from once living organisms. It is most useful for samples up to roughly 50,000 years old. Beyond that, the remaining carbon-14 signal becomes difficult to measure reliably. Greenland Shark research uses radiocarbon in a specialized way, leveraging the bomb pulse calibration created by mid-20th century atmospheric weapons testing. This calibration allows precise dating of tissues formed during or after that period and supports growth modeling for older specimens. Genetic modeling supports age estimates for organisms like Pando and Wrangel mammoths. By measuring mutation rates and population structure, researchers infer how long lineages have been separated or how long clones have existed. Genetic clocks produce ranges, not exact birthdays. Stratigraphy underlies dating at fossil forest and stone tool sites. Volcanic ash layers, sediment sequences, and magnetic reversal signals can bracket ages by establishing that a sample lies between two well-dated horizons. The Springfield fossil forest sits within a broader stratigraphic framework that ties it to the Pennsylvanian Carboniferous. Lomekwi sits within a stratigraphic sequence anchored by volcanic horizons. Direct dating measures the object itself. Contextual dating measures material above, below, or around the object. Both are valid, but they answer different questions. Direct dating is preferred when possible. Minimum ages are common in cave art and some fossil contexts. They establish that something happened before a known cap without specifying exactly when. Uncertainty ranges such as plus or minus 8 million years for Jack Hills. Zircons, or plus or minus 2.2 thousand years for Sulawesi. Minimum dates do not make a date meaningless. They quantify the precision of the measurement, allowing comparison with other dates and with the broader timeline. Better instruments can push timelines older without changing the object. The same crystal, the same painting, the same fossil can return refined ages as methods improve. Every dating method depends on assumptions about system closure, contamination, and sample integrity. When those assumptions are violated, dates become unreliable. Cross-checking with multiple methods is the standard response. The past becomes measurable when chemistry, physics, and context agree. Earth is not a museum. It is an active recycling machine. Crust melts, sinks, and is reformed. Mountains erode into sediment. Sediment compacts into rock and is buried, exhumed, or subducted. Plate tectonics drives the entire system. Most of Earth's deep history is destroyed by ordinary geological processes operating on long timescales. The Jack Hills Zircons exist because their parent rocks were eroded, but the Zircons themselves were durable enough to survive transport, burial, and re-exposure. The original Hadean crust is gone. Biological decay erases soft bodies quickly. Ediacaran organisms and Francevillian structures, if biological, survive only because exceptional preservation conditions intervened. Microbial mats, fine sediments, low oxygen seafloor environments, and periodization all played roles. Such conditions are rare and patchy. Most early life left no trace. The Springfield fossil forest exists only because rapid burial interrupted decay. Most ancient forests were not preserved. They rotted in place, their carbon recycled into atmosphere, soil, and ocean. Standing forest preservation requires earthquake-driven subsidence, volcanic mudflow burial, or comparable events. These are not background conditions, they are accidents. Archaeological loss follows similar patterns. Stone tools survive far better than wooden ones, leather, fiber, or food residues. The behaviors and intentions surrounding the Lomekwi tools are not recoverable. The maker's social structures, communication systems, and cognitive worlds disappeared. Pigment loss in caves is an active, ongoing problem. Maltravieso and Sulawesi survived because cave chemistry, humidity, and mineral crusts protected pigment for tens of thousands of years. At Sulawesi, climate change and seasonal humidity shifts are now causing exfoliation, the flaking off of pigmented surface layers. Some of the earliest evidence of human storytelling may be deteriorating faster than it can be documented. Genetic loss compounds the problem. Extinct populations leave fragmented DNA, often only recoverable from permafrost or other cold environments. Wrangel mammoths are studied genomically only because Arctic permafrost preserved their tissues. Most extinct species offer no comparable opportunity. Living loss is also real. Organisms like Pando can survive while their earlier ecological context disappears. Pando still grows, but the climate, fauna, and ecosystem dynamics that shaped its early millennia are gone. Modern pressures, including grazing, drought, and altered fire regimes, can erase ancient biological continuity within decades, faster than any geological process. The mainstream principle is that most deep-time information is destroyed by normal Earth processes. The historical angle is that mining, erosion, cave exploration, and accidental exposure often reveal what nature had hidden. Discovery itself is uneven and opportunistic. The more speculative reading is that many lost worlds may be permanently unrecoverable. They were not impossible. Their evidence simply failed to survive. Modern relevance is urgent. Preservation is rare and fragile. The same ecosystems that produce future fossils are the ones being altered most rapidly today. Deep time is not hidden by mystery. It is erased by ordinary physical law operating across scales humans rarely feel. These 10 anomalies warp our sense of time because human history is too short to provide a useful frame of reference. Earth preserves time unevenly, not conveniently. Minerals can outlive continents, as the Jack Hill Zircons demonstrate. Fossils can preserve vanished body plans, as the Ediacaran Biota show. Coal can turn forests into geological architecture, as the Springfield site reveals. Tools can survive after their makers disappear, as Lomekwi makes clear. Clonal roots can outlast civilizations, as Pando demonstrates in real time. Pigments can preserve symbolic behavior across species, as Maltravieso suggests. Cave scenes can preserve early storytelling, as Sulawesi shows. Island bones can place Ice Age animals beside Bronze Age history, as Wrangel mammoths place beside the pyramids. Living sharks can carry centuries inside a single body, as the Greenland Shark demonstrates beneath the Arctic ice. None of these discoveries require exaggeration to be powerful. The technical facts already create the wonder. 4.4 billion years inside a sand grain-sized crystal, 2.1 billion years for possible early complex life. Animal-defining cholesterol in a fossil more than 500 million years old. A fossil rainforest preserved in the roof of a working coal mine. Stone tools made before the first member of our genus walked the planet. A single living organism that may predate the last ice age. Hand stencils painted by a different human species. Hunting scenes painted before Europe had any equivalent. Mammoths alive when the pyramids were under construction. A shark possibly older than the telescope. The strongest approach is to respect uncertainty rather than hide it. Science often becomes more interesting when dates are ranges rather than absolutes, when interpretations are debated rather than settled, and when each new dating method opens questions that previous methods could not address. The mainstream closing idea is that deep time is reconstructed through evidence methodically with constant refinement. The historical closing idea is that human civilization is only the latest thin layer on a much older planet. The speculative closing idea is that future methods may yet reveal even older traces hiding in plain sight, in samples already collected, in caves already mapped, in fossils already on museum shelves. Innovation in nature comes from simple laws repeated over impossible durations: radioactive decay, crystal lattice physics, cell division, photosynthesis, erosion, burial, mineralization, mutation, selection. Each is straightforward in isolation. Each becomes profound when run for millions or billions of years. Time does not move faster or slower for the planet. Humans simply arrived very late, equipped with brains that evolved to track seasons rather than eons. The anomalies in this documentary are not anomalies to Earth. They are anomalies only to us. The planet has always been keeping its records. We are just now learning how to read them.